Hello everybody, today is my presentation and my topic is variation of foliar defensive compounds and fungal endophytes abundance among different locations in mature white spruce. Before we go into our study, here is my personal information. My name is Haolin and I'm a second year PhD student in the Deers Lab. My research topic is interaction between different fungal species. And here is an interesting picture to hook you in my research question. What are the black dots on needle? Do you think it become harmful? You will know the answer at the end. So our study is related to fungal endophytes, tree defense compound terpenes, and their relationships. So here are two objectives. First, we want to investigate the variations of fungal endophyte abundance and terpenes among 6 study sites. Then we want to finding the correlations between fungal endophyte abundance and terpene concentration. Based on this, we have 6 different study sites among 5 different provinces, including Alberta, Saskatchewan, Ontario, Quebec, and New Brunswick. And here is the map, shows our study sites. And on the left part is our data table. We collect some information such as province ID, latitude, longitude, and elevation of each study site. Each study site, we choose 5 trees and they are all 20 to 30 years old mature white spruce with a DBH larger than 20 centimeters. DBH is about 1.3 meters high, and we collect both foliar tissue and phloem tissue. Foliar tissue, we cut them with a paw pruner, and you can see in the left side picture. For the phloem tissue, we use the hollow leather punch to put inside the bark, and the right side picture it shows the hollow leather punch and the piece of phloem samples. And then we analyze these 2 samples in 2 different machines. For foliar, we based on their DNA products and use 2-step PCR plus sequence A using the right side picture Eppendorf thermal cycle here. For the phloem, we use chemical analysis with GCMS in the left side picture here. After analysis, we have some key results. First of all, we found significant variations in fungal endophyte abundance. And you can see here is a group bar chart with y-axis is abundance of each fungal endophytes. The unit is ASVS. You can understand is that higher ASVS means more abundant of that fungi of that endophyte genera. And x-axis is our location. So you can see here, Alberta and Saskatchewan have higher endophyte abundance, while the dark pink bar is the cladosporium. It is the most abundant. It occurs in every 5 locations. And after that, we found no variation in terpenes diversity. And you can see here is the same bar chart. The y-axis is actually the concentration of each each terpene compounds we found in our study, and the axis is our location. So you can see each location they have similar diversity of chemical terpenoid compounds. So there's no variation. After that, we found strong positive correlations between 4 fungal endophyte genera and total terpenoid concentration based on the PCOA plot. And here the plot shows the correlations between different vectors, and the vectors is the arrows. And here we concentrate on the arrows in this arrow. So you can see Larula, Charisomesa, Chelara, and Calendospermides. These 4 genera shows very small angles with the monoterpene and sesquiterpene, means they are stronger correlated with each other. And this finding gave us a gateway for new biological control agent for spruce spotworm, because monoterpene sesquiterpenes are defined can be reduced the development of spruce spotworm, while some endophytes can induce their biosynthesis of the trees, but no studies found white spruce focus on the white spruce species, and here our study gave a key finding of 4 ideal fungal endophyte genera in white spruce trees that can be used as a potential biological control agent for spruce spotworms in the future. Finally, we found there is no correlation between any detected terpene concentration and total endocyte abundance, and it's the same PCOA plot with the last one. And we also focus on the errors, which is the vectors. You can see the total endocyte vector lies in the other side of the picture, so it means that there is no correlations with total endocyte concentration with any other terpenoid compounds we found. Based on this, I have a conclusion here. First, we found large variations in fungal enophytes abundance in white spruce across Canada, and this finding provide us essential knowledge and information for spruce enophyte fungi distribution, because enophyte is actually a new species which less than 100 years study, and nobody actually gives a very clear distribution of different abundance of different fungi genera and fungi enophyte genera across Canada on white spruce. So our findings can provide very meaningful knowledge on that, which can give us a baseline of biodiversity for future forest management practices. Mm, Moreover, we found endophyte Lerula, Charosimisa, Charala, and Clindosporidae include induced biosynthesis of terpenes in white spruce, and this provides a gateway for using endophyte as a biological control agent, as we see in the last slides. So finally, back to our question at the very beginning, you can see actually the black doors are fungal endophytes, and according to our study, they don't actually be harmful for the host. Instead of them, they can help them to produce more defensive compound, monoterpene and sesquiterpenes, and maybe become a potential biological control agent for the spruce spotworms in the future studies.